For part two of the chemistry review, we're going to start with biochemistry. So biochemistry, so this includes chemicals that can be either organic or inorganic. The inorganic compounds do not contain the element carbon. So that includes things like water, salts, acids, and bases. The organic compounds, they contain carbon and are usually large and have covalent bonds. So that's things like carbohydrates, fats, proteins, and nucleic acids. Water is the most abundant inorganic compound, so it makes up 60 to 80% of the volume of a cell. It's one of the most important inorganic compound because of the properties of water. Uh, it has high heat capacity. That means that it can absorb and release heat with little temperature change. So it prevents sudden changes in temperature. So that's one of the reasons why places that are more inland or more in the desert area, you can see more drastic temperature changes. So if it's hot, it'll get really hot, but when it gets cold, like at night, it'll get really cold because of the absence of water. Compared to more coastal areas where the temperature is more moderate, or mild. It also has high heat of vaporization, so that means that evaporation requires a large amount of heat. Okay, so it's a useful cooling mechanism. It has polar solvent properties because it dissolves and dissociates ionic substances, and reactivity because it's a necessary part of reactions like hydrolysis and dehydration synthesis. And then finally, cushioning because the water can provide protection to the body and some of the organs. So, for example, the cerebral spinal fluid does provide some protection for the brain. So here you can see how the dissociation of salt and water, because salt is an ionic compound, so water is very effective at dissolving ionic compounds. And having an ionic balance is something that's vital for maintaining homeostasis. Some of the common uh, salts that are found are like sodium chloride, calcium carbonate, potassium chloride, and calcium phosphates. So a lot of these are important for things like nerve impulse transmission and also muscle contraction. Like calcium and sodium, they open a lot of those channels that allow for things like muscle contraction. Acids and bases, so both of these are electrolytes. They ionize and dissociate in water. Acids, they release hydrogen ions, okay, and bases, they pick up the hydrogen ions in a solution. So in an ac- in an acidic solution, there's going to be more hydrogen ions, whereas in a basic solution, it's going to be less. There's going to be less hydrogen ions. Okay, and that's related to the pH scale. So as you can see here, in the pH scale, well, the pH is a measurement of concentration of the hydrogen ions. So anything that's very acidic will have more hydrogen ions. Anything that's basic would have less hydrogen ions, and it'll have a higher pH. Buffers are things that can resist abrupt changes in in the pH uh, in pH levels. And that's because they can release hydrogen ions if the pH rises. So if they release hydrogen ions, that's going to increase the acidity, so it'll bring that pH level down. And they can also bind hydrogen ions if the pH falls. So if the pH falls, that means that something is very acidic. So there's a lot of hydrogen ions, and by binding those hydrogen ions, it'll decrease the number of hydrogen ions. So it'll bring that pH level up. Carbohydrates include sugars and starches. So there's three classes of carbohydrates. There's monosaccharides, which are single sugars. So that's things like glucose, ribose, right? Ribose is a sugar that's found in RNA, and there's deoxyribose, which is the monosaccharide that's found in DNA. Then there's disaccharides. So these are two sugar groups. So that's things like sucrose, lactose, and maltose. And that's two sugars because, like, for example, for sucrose, that's having glucose and fructose. So it's fructose plus glucose. That makes sucrose. So yeah, so that's why it's a disaccharide, having two sugars. Polysaccharides have several sugar groups. So that includes things like starch and glycogen. Starch is um, the main way that plants store energy, and glycogen is the main way that humans store energy. So glycogen is when you have excess sugar in your body or excess glucose that's not used, the liver, it'll a lot of it will be stored in the liver as glycogen. For lipids, there's triglycerides. So that includes saturated fats. Saturated fats, in these, all the carbons are linked by covalent bonds. So it results in a molecule with the maximum number of hydrogen atoms, and so it's referred to as being saturated with hydrogen. So it's called a saturated fat. So these are solid at room temperature. So this things like animal fats and butter. Unsaturated fats, so these have one or more carbons that are linked by double bonds. So it results in having a reduced number of hydrogen atoms. So it's called unsaturated. And there's these double bonds can cause a kink in the fatty acids. So that means that they can't pack close together or as close together as things like these saturated fats. And this allows them to be liquid at room temperature. So for example, the oils that you should be familiar with, like plant oils, uh, olive oil, things like that. And then there's trans fats, which are modified unsaturated fats, and these resemble the structure of saturated fats. These are the ones that are considered to be unhealthy. The healthy fats are things like omega-3 fatty acids. Then there's other things like phospholipids. So phospholipids are something that you usually see um, forming involved in some type of layer. So you probably have heard of like a phospholipid bilayer. So it's a two layers of phospholipids, like the one you see here in this diagram. So they have two main regions. So there's the head and the tail regions, but they both have different properties. The head is polar, and it's going to be hydrophilic. So that's going to be the part that faces the water. And then there's going to be the tail, which is nonpolar, and that's going to be the hydrophobic region. So it faces away from the water. So that's why it makes it an effective protective layer because it prevents water from penetrating. So it keeps the water out. And then there's steroids, which consist of four interlocking ring structures. One of the most important steroids would be cholesterol, because cholesterol is the starting material for the synthesis of things like vitamin D, steroid hormones, and bile salts. Bile salts are lipids that are produced by the liver to break down proteins. All proteins are made of 20 types of amino acids. They're joined by covalent bonds that are called peptide bonds, and they contain both the amine group and the acid group. There's 4 levels of protein structure, and these determine the shape and the function of the protein. So there's the linear sequence of amino acids. These are just the chain of amino acids, okay? Like the ones we were looking at in lab when we were putting together the RNA strands with the amino acid groups. Then there's the secondary structure, which is how the primary amino acids interact with each other. So that includes the alpha helix and the beta plated sheets. Okay. And then there's the tertiary structure. So that's how the secondary structures interact. And then the quaternary structures, how 2 or more different polypeptides interact with each other. So here you can just see the diagrams. So we have the linear structure, which is the primary structure, and that's just a chain of amino acids. The secondary structure includes the alpha helix and the B sheets. Okay. And then you have the tertiary and the quaternary structures. For protein denaturation, so this varies based on the type of protein because there's globular proteins and there's fibrous proteins. The globular proteins, if they're denatured, they do lose their um, their form and their function. So without the form, it'll have it can't conduct its specific function. The fibrous protein, these are more stable, so they're less likely to, or they don't denature as easily as the globular proteins. So they have more of a structural function than globular proteins. Um, Fibrous proteins, so this would include things like actin and myosin, which are proteins that are found in muscle cells. There's keratin, right, a protein that makes up things like your hair, your nails, and then there's elastin, which is found in connective tissue. Globular proteins, that includes things like enzymes. Enzymes are globular proteins, and also insulin. So the denaturation of these proteins can be attributed to a decrease in pH or an increase in temperature. So usually it is reversible if the normal conditions are restored, but if the conditions are too extreme, then it is not reversible. For example, when you cook an egg, I mean, once the egg is, it's fried or heated to a certain degree, it's going to change in the form of the egg is going to change completely. So that cannot be undone. Enzymes. So like I mentioned, these are type of globular proteins. So these are the ones that act as biological catalysts. So they regulate and increase the speed of chemical reactions without getting used in the process. So the main function of enzymes is to lower the energy that's needed to initiate a chemical reaction. Okay. And when you lower the energy that's needed, that's going to lead to an increase in speed of the reaction and allows for multiple reactions to occur or many reactions to occur. That's because the enzyme it binds to the substrates and then it brings them into the best position and it brings them into the best position that's needed for reacting for a reaction to occur. So the activation energy, so like I mentioned, enzymes do lower the activation energy, which is the energy that's needed to initiate the chemical reaction. And this happens in 3 steps. So first, the substrate is going to bind to the enzyme's active site. and it's going to temporarily form an enzyme substrate complex. Then the complex is going to undergo a transformation, and then eventually the product is going to be released by the enzyme. Okay, so here you can see two diagrams that shows the difference between a reaction that happens with an enzyme and a reaction without an enzyme. Without an enzyme, it requires a lot more energy, so more activation energy is required for that reaction to occur. Whereas with an enzyme, the reaction requires less energy. So again, it's just more efficient, okay, because the enzyme puts the substrates into the best position to react with one another, and so it requires less energy for the reaction to occur. And here you can see those steps in the enzyme action. So you have the enzyme with the active site. It'll bind to substrates. It forms the enzyme substrate complex. Then this is where the bond is formed, and you have the storing of absorbed energy. And then eventually, once the reaction takes place, the product is separated from the enzyme, and then um, the enzyme is able to bind to another substrate. Nucleic acids are made up of nucleotides. So these include a nitrogen base, the pentose sugar, and a phosphate group. And there's two major classes of nucleic acids. So there's the deoxyribonucleic acid, which is DNA, and then ribonucleic acid, RNA. So DNA holds the genetic blueprint for the synthesis of proteins. It's double-stranded, right? There's it has the double helix with the two strands. And the nucleotides contain deoxyribose sugar, the phosphate group, and one of the four nitrogen bases. So we looked at the nitrogen bases in lab. So there was adenine, guanine, cytosine, and thymine. So the way that these nitrogen bases bind is very specific. So they bind to the strand the that's opposite to one strand, and it binds to a specific base. So adenine will always bind with thymine. You have guanine that will always bind with cytosine. Okay, so this is what it looks like, how those bases bind to a specific base on the opposite strand. RNA is what links DNA to protein synthesis. So it's the intermediate step that has to take place. And it's slightly different than DNA. So instead of having a double strand, you have a single strand of a linear molecule. And this is going to be active outside of the nucleus. So if you recall, DNA is active inside of the nucleus. RNA is more active outside when it's in the cytosol. And RNA contains ribose instead of deoxyribose as the sugar group. And then thymine is replaced with uracil. So there's three varieties of RNA that carry out the DNA orders for protein synthesis. There's the messenger RNA, which is the initial strand that's generated from the template strand of DNA. And then there's the transfer RNA that brings the amino acid um, towards the ribosome. So as the ribosome is reading those codons, so those three uh, bases of the nucleotide, when it's reading it those three letters, three at a time, then depending on the sequence or the letters that are being read, the transfer RNA will bring the transfer RNA with a specific anticodon will bind, and it will bring the amino acid with it, okay, because it's part of it. And then there's a ribosomal RNA, so that's involved. That's the main component in ribosomes, which is the organelle that's reading those um, base sequences, so those codons. ATP stands for adenosine triphosphate, and it's essentially the energy currency that the body uses to get things done because it powers the chemical reactions in cells. So it provides the usable energy that's needed by the body cells. It's made up of the adenine containing RNA nucleotide with two additional phosphate groups. So a total of three phosphate groups. That's why it's called triphosphate, adenosine triphosphate. It could be in various forms. So if it loses one of those phosphate groups, then it becomes ADP, which is something that you'll also hear about. So this will conclude chapter 2, part 2. This is what you have to know for the upcoming quiz, okay? Because the quiz is going to be on chapter 1 and chapter 2. As far as the sections that we went over in part 2, for chapter 2, it was 2.6. We didn't do 2.7, so we're skipping that. And then it's 2.8 through 2.12.